Hey guys, Miss Zimmerman here. Today we're going to be talking about organic chemistry. Make sure you have your Cornell notes set up in your notebooks. The lecture today, pink is going to be our headings, blue is vocabulary, and yellow is important words. I want you to make sure that you highlight the vocabulary and the important words in your notes, two different colors. You might want to label your vocabulary yellow and then your important words in orange. All right, so what is organic chemistry? This word right here, organic, tells us a very important thing, that it contains carbon. So any compound on the planet that contains carbon is considered organic. And we are considered organic life beings because we contain carbon. Now, if you were looking for carbon on the periodic table, you would look for number 6. Carbon has 6 neutrons, 6 protons, and 6 electrons. Where would you find these two in the atom? If you picked the nucleus, you'd be correct. And then the electrons are found on the electron orbitals. Now, if you look, here's our outside orbital, and there are 1, 2, 3, 4 electrons in the outside orbital, which tells us that we have 4 valence electrons, and this allows carbon to make 4 bonds. Now, if we were to look at the human, because that's what we're really concerned about, or animals or plants, carbon, hydrogen, oxygen, and nitrogen, those are the 4 elements that make up the majority of the mass of a human, 96% to be precise, and that's what we call natural elements. Now, humans have what we call trace elements, which are only needed in very small amounts, and we usually call these minerals. All right, so the branch of organic chemistry that we're going to be focusing on in biology is looking at the macromolecules. There are 4 major macromolecules in the human body, and they are going to essentially make up everything that is important and what we will be talking about this year. Now, macromolecules are built by these things called monomers. You could think of the macromolecule as a house, and then the monomers would be the bricks that make up the outside, the paint that you're painting the walls, the flooring, the roof. Those are your building blocks. And those building blocks are going to link together and make these molecules called polymers, which is just another name for a macromolecule. And that process is called polymerization. Now, to form these polymers by putting together monomers, that reaction is called dehydration synthesis. It's also called condensation reaction. And if you look at this word, dehydration, you've seen this word before. What does it mean? It means to take out water. You know, we have dehydrated food, or if you're going to dehydrate meat to make deer jerky, you're taking out the water. So to form these polymers, you have to remove a water. So for example, we have a 2 monomer unit right now, and we want to add on a 3rd. So what we're going to do is take the hydrogen from this end and the hydroxyl group from this end and form water. And by taking these 2 things out, we can form a bond between this one. And now we have a polymer that is 3 monomers long. So if we wanted to add another monomer onto this side, what would we have to do? You would have to take this hydrogen out and the hydroxyl group of another. We would form a water, and then we would be able to put a bond between there. Now, what if we wanted to break a bond that we formed? We wanted to digest our food and get the energy that's stored within these bonds. That process is called hydrolysis. And in order to separate the monomers or break these bonds, we have to add water back in. So that process is pretty simple. You take water, you put it back in, we're going to break that bond, and now we're going to have a hydroxyl group right here and a hydrogen group right here. If we wanted to break this bond, we would have to add a water in there and break it. Super simple. 2 major reactions to form and to break for all 4 of our macromolecules. So, 4 macromolecules we're going to be looking at are carbohydrates, nucleic acids, lipids, and proteins. Don't worry about these page numbers. We are going to be using a different text, so just ignore those. But the 4 major macromolecules are carbohydrates, nucleic acids, proteins, and lipids. I want you to pause, and I want you to make a little drawing for each of these, what they look like in this picture. Okay, now that you have your drawings done, let's talk about each of the 4 macromolecules. The first one we are going to talk about is carbohydrates. It's made up of C, H, and O. What are those 3 elements? Carbon, hydrogen, and oxygen. Great. So the whole reason we consume carbohydrates or have carbohydrates in play is for energy. Now we have 2 kinds of energy. We have the first kind called sugars. Those are going to be really quick sources of energy. Think about candy and candy bars and those really sweet things. Those are going to contain carbohydrates or sugars like glucose, lactose, fructose, and sucrose. And those are super small, so we're able to break those down really easy and get a quick burst of energy. We have these things called monosaccharides. Those are our monomers. Those are going to be our building blocks to make more complex carbohydrates. Monosaccharides are simple sugars. They cannot be broken down into smaller pieces. They are the building block. Those are going to be things like fructose, ribose, deoxyribose, glucose. We'll be seeing these words a lot this year. Just remember that those are our monosaccharides. And if you remember mono means 1, you know that that's 1 unit. Now a disaccharide, we could kind of figure out what that means by the prefix here, which is di. Di means 2. So all we're doing to form a disaccharide is putting together 2 monosaccharides. For example, sucrose and lactose are 2 different disaccharides. And then polysaccharides is another kind of sugar that we can make. Poly means many. So in a polysaccharide, we're putting together many different monosaccharide units. Cellulose, starch, and glycogen are 3 of the main polysaccharides we'll talk about. Cellulose is found on the outside of plants, and it is not digestible by humans. So think of corn. What happens when you eat corn? You can't digest it. So that is because of the cellulose outside layer. Starch is the storage form in plants, and we can digest that. That's where we're going to get our energy when we're eating potatoes and French fries. And then we have another thing called glycogen, which is the storage form for humans. And we store this polysaccharide in our liver. The other kind of carbohydrate we have is starches, which is a polysaccharide. It has many sugar units linked together. It's a great source of energy. However, it's going to break down slower. Potatoes, pasta, bread, grains are the main kind of starches that we'll see. And think about people that are runners. Have you ever heard of carbohydrate loading? If they're running a marathon, the day before they're going to eat a ton of these starchy foods because their body is going to break it down slower, and they will have energy for the next day for their very long run. Now, if we look at the structure of a starch, these are their monomer units, In this case, it's glucose. So we have a whole bunch of glucose molecules bonded together. Now, if we wanted to break this bond here and break down the starch that we've just eaten to get the energy from it, we would have to go through hydrolysis. And what would we have to add in here to break that bond? If you said water, you'd be right. All right, the next macromolecule is a lipid. That's going to be mainly made up of carbon and hydrogen. They are used to store energy for long term. They're used for insulation and protection. They're going to be our fats, our oils, our waxes. Think about humans. We are going to store the energy we don't use from our food in the form of a fat, which is a lipid. They're what we call insoluble in water, which means they don't dissolve. So think about if you pour oil in water when you're making pasta. That oil is going to stay at the surface of the water and it's not going to dissolve. So that is one of those main characteristics of these lipids, that it's not dissolvable or not soluble in water. Now, there are two kinds of lipids. You have saturated lipids and you have unsaturated lipids. To tell the difference between the two, saturated lipids are going to form a solid at room temperature. Unsaturated are going to stay liquid. So I want you to pause and make a list of some things that you know that are going to be saturated fats and some of the things that are going to be unsaturated fats. All right, if I was making that list, my saturated fats, I would probably say bacon grease, Crisco, the grease from your hamburgers. Okay, saturated fats are usually our bad fats or our unhealthy fats. Unsaturated fats that stay liquid at room temperature are going to be our olive oil, our vegetable oil. Now, the reason that those behave the way that they do is that unsaturated fats have this kink in their structure, and that kink allows them to stay liquid because they can't pack together really well, whereas saturated fats don't have this kink. So we have three straight fatty acid tails here, and they're able to pack together really well, so they're going to solidify at room temperature. Now, if we were looking at a lipid, there's really two major parts or components that make it up. You have this part here, which is called the glycerol, and then you have these, which are fatty acids. All right, proteins. Those are made up of nitrogen, carbon, hydrogen, and oxygen. Their monomer or building block is going to be an amino acid, and we have 20 of those in nature. Now, proteins are the biggest, broadest, most complex category, which is kind of strange because there's only 20 things that make them up. But how we put those 20 things together and how big we make those proteins is going to affect what those proteins do. So they have a large variety of sizes and shapes. If we change the shape of the protein, it's going to become a new protein, which means it's going to have a different function. So if you change the shape, you change the function. For example, when you boil an egg, the protein changes color and shape. That is now a new protein, or it serves a different function. And proteins are pretty much found everywhere, from muscles to the cell membrane. All right, so proteins are going to be made through the process of dehydration synthesis. You're going to be adding amino acids together and to form a chain. And we're going to be talking about this process of protein synthesis in detail because it serves a very, very major role in all of our biological processes. But if you know for just now that the building blocks are amino acids, they're going to be built through dehydration synthesis and broken down through hydrolysis, you'll be okay.